Hello friends, welcome to my YouTube channel Neat Biology Made Easy. In this video, I'm going to discuss on proteins. But before I start, I request you to kindly subscribe my channel, press the bell icon for regular notifications. My last video was on amino acids and you can find the link in the description section. So amino acids are the building blocks of protein. So here I have shown you the structure of an amino acid. So valency of carbon is 4. So one of the valencies are occupied with the amino group, and here the other valency is occupied with carboxyl group. And the third valency is occupied with hydrogen, and the fourth valency may be occupied with any functional group which may be designated with R. So then the resultant molecule may be considered as an amino acid because it bears both amino group NH2 and acid group that is COH. Here I have shown you two amino acids with adjacent carboxyl group COH and NH2 group. So due to the release of water, it forms peptide bond, and this reaction is called condensation reaction because it in this reaction water molecule is released. Then one very important point that we should remember for entrance examination is that peptide bonds are covalent bonds and are strong bonds and are responsible for the stability of protein structure. Then the second important point to remember for entrance examination is that peptide bonds are planar. So here in this diagram, I'm showing you two spheres connected with one stick. So obviously, if two spheres are connected with just one stick, then the rotation around this stick is possible. So that is why these two actually atoms may not be existed in the same plane because rotation is possible. So they, due to the rotation, they may not exist in the same plane. So the same thing should also happen with C and N because they are connected through only one bond. But such things won't happen in case of peptide bonds because in peptide bond we can see partial double bond character between carbon and nitrogen, and this is due to the delocalization of electrons in C double bond O and C bond N of the peptide group, and the partial double bond characters may be seen between C bond N, which prevents the rotation, and that is why peptide bond exists in planar form. So in this diagram, you can see if two spheres are connected with two sticks, obviously rotation around these two sticks won't be possible, due to which this kind of molecule uh, will exist in planar form, and that is why peptide bonds are planar. So next is classification of protein. So based on physicochemical properties of protein, they may be classified into two groups: water insoluble protein, also known as fibrous proteins. So here the examples are meat, collagens of muscle, then plant fibers, keratin protein, which basically makes our hairs, nails, and horns and hoofs in animals. All these are fibrous form of proteins and are water insoluble in nature. Then comes water soluble protein, which are mostly globular proteins. So basically, saliva, egg albumin, alpha and beta globulins are the etc. are the examples of water soluble protein. Based on structural complexity, protein structures may be categorized into four groups: primary, secondary, tertiary, and quaternary structure of protein. So let's discuss on primary structure of protein. So primary structure of proteins are nothing but linear chain polypeptides. So here I'm showing you one example of linear structure protein, and that is insulin. So insulin is a hormone which is produced in the beta cells of pancreas in human body, and they helps in metabolizing glucose. So here I have shown you the chemical structure of insulin. So it has got two linear chain. One is called A chain and the second one is B chain. So A chain is consist of 21 amino acids, and the B chain bears 30 amino acids, and these two chains are connected with each other by disulfide bonds, and these disulfide bonds are formed between cysteine. In my last video, I forgot to mention that cysteine and methionine are the sulfur containing amino acids, which helps in the formation of disulfide bonds in proteins. So disulfide bonds also help in protein folding, and they also provides stability to the protein. Then secondary structure of protein. So again, secondary structure of protein may be subclassified into two group. One is alpha helical structure, and second one is beta pleated sheet structure. So in alpha helical structure, we can see the formation of hydrogen bonds between C double bond O and NH groups within the same polypeptide chain. So here on your right hand side, I'm showing you one helical structure of protein. This is the same linear structure. Actually, it forms helical structure due to the formation of hydrogen bonds between C double bond O and NH group. So again, there may be two possibilities of in alpha helical structure. We may have both left-handed helix, then again right-handed helix. So one example of alpha helical protein is transmembrane protein. So if you see the structure of fluid mosaic model in your textbook, you'll see the presence of transmembrane protein in the fluid mosaic model. So these are all actually alpha helical form of protein. Beta pleated sheet structure. So beta sheets consist of beta strands. They are again connected laterally by at least two or three hydrogen bonds between C double bond O and NH groups. So they are also found in some of the membrane associated proteins. So here in this diagram, I'm showing you both alpha helical presence of alpha helical structure in the fluid mosaic model, and of course the beta pleated sheet like structure in the same fluid mosaic model. And here in this diagram, I have shown you the comparison among various forms of beta pleated sheet structure and the alpha helical structure. So on your extreme left hand side, you can see uh, there's a single polypeptide beta pleated sheet like structure. So within the same polypeptide chain, hydrogen bonds are formed. So if hydrogen bonds are formed within the same polypeptide chain, then we call them intramolecular hydrogen bond. So in beta pleated sheet like structure, we have both the possibilities. That means either we may have intramolecular hydrogen bonding, and also we may have intermolecular hydrogen bonding. So in this diagram, you can see there are two polypeptide chain actually, and they are connected with hydrogen bonds. So as these hydrogen bonds are formed between two different polypeptide chains, so we call them intermolecular hydrogen bonding. But again, in alpha helical structure, you can see the formation of hydrogen bond within the same polypeptide chain. So one more point which is very very important for entrance exam is that Alzheimer's disease. So one of the reasons behind Alzheimer's disease is the refolding or misfolding of beta amyloid protein, which is endogenous to human brain tissues and which may lead to Alzheimer's disease. And this point is important. Then coming to tertiary structure of protein. So tertiary structure is the overall three-dimensional structure of a polypeptide chain, which is again actually formed with the help of different forms of secondary structure. They assemble together and they forms three-dimensional protein structure. So the tertiary structure is primarily due to the polypeptide side chain interactions between the R groups of the amino acids, and they make up the protein. So one such example is ribonuclease enzyme, and this example is important from exam point of view because they may ask to write one of the examples of tertiary structures of protein, and ribonuclease enzyme is a form of tertiary structure of protein. And I have mentioned two more examples, and these are triose phosphate isomerase enzyme and bacterial class 2 HMG coenzyme A reductase. Here in this diagram, I'm showing the tertiary structure of triose phosphate isomerase. You can see the presence of both alpha helical and beta sheet structure. So here in this diagram, the beta pleated sheets are designated with these solid arrows. Then comes quaternary structure of protein. So again, quaternary structure of protein is a three-dimensional structure of protein, but it is formed due to the presence of two or more polypeptide subunits in three-dimensional arrangement. So one such example is hemoglobin, which consists of two alpha and two beta chains. So here in this diagram, you can see the presence of two alpha helical structures and two beta sheet-like structures. So for your reference, I have drawn this diagram on your right hand side. I have used two alpha symbols and two beta symbols. Together they makes up the quaternary structure of protein. And this diagram will help you to memorize this concept for your entrance exam. So again, there may be some of the questions which can be asked based on the differences between hemoglobin and myoglobin. So basically, myoglobin is found in muscles in mammalian body, and of course in some of the insects. Then hemoglobin is present almost all over the mammalian body. And one most important difference between hemoglobin and myoglobin structure is that hemoglobin structure is heterotetrameric. That means hetero means different. That means it bears both alpha and beta chains. And tetrameric means four. Tetra means four. So it has got two alpha and two beta chains. So total four subunits are there. Then myoglobin is a monomeric structure. That means it bears only one polypeptide chain. Hence it is called monomeric structure. So here I have listed some of the important points that we should revise before appearing for any entrance exam. So first point is polypeptide bonds are covalent bonds and they are strong and they helps in protein stability. Second is the insulin is a primary form of protein which is composed of A and B chain. Then again 21 amino acids are present in A chain and 30 amino acids are present in B chain, and they are linked together by disulfide bond. Then hydrogen bonds formed between C double bond O and NH groups in polypeptide chains and are responsible for secondary structure of proteins. Then disulfide bonds are responsible for protein folding and stabilization. One of the reasons behind Alzheimer's disease is misfolded proteins, and this point is really important from entrance point of view. Then the structure of hemoglobin, it's a heterotetrameric structure with two alpha and two beta chains, whereas myoglobin is a monomeric structure. So thank you so much for watching my video. Please like, subscribe, and share my videos. Thank you.